for the need for clinical trials. Why do we want clinical trials? Why do we need? And why there is a difference between association, causation, and other problems that come about when we study relationships. And we'll talk about evidence creation and what we mean by PICOT. But it's a way of characterizing, describing the research on us. So the reason we want clinical trials, we need clinical trials, that we as humans, all of us, we are interested in what are the causes of clinical conditions. So you cannot see the slide detailed, but this is a causal loop diagram demonstrating the complexity of the COVID-19 pandemic, environmental, health, socioeconomic system. So there's all sorts of things from social aspects to economic aspects, environmental, health, and then personal reasons. And our beliefs, our condition, all of these things are related to economic factors, related to our health and to the environment. Things are very complex. And things are very complex. Is that okay? That's better. Very complex thing. Okay. So we want to know nothing is simple. Reality is very, very complex. Relationships are very, very complex. Not only human relationships are complex, but relationships in the health field are very, very complex. What we try to do is to simplify things, right? Because what we're interested in is why, let's say, I get long COVID, for example. Why do I get it? So I may be interested in why it occurs, when it occurs. Does it occur after certain things, under certain circumstances? How severe does it occur? And what is the cause of its cure? The reason we want to know it is so that we can either prevent it, we can treat it, or we can cure it. And so this is the example with COVID, but you can take any example: renal failure, heart attack, miscarriage, whatever. Good things, bad things you could take. Somebody getting cured from cancer, for example. So we want to know these things. So first thing is we are interested in knowing causes. And we want to then establish the evidence for this. And you may have heard, and this actually, I got this out of a textbook, but I put the different colors in it. So randomized controlled trials. Oh, and that's one thing to clarify. RCT, we tend to think it's randomized clinical trials. No, the C is not for clinical. It's for controlled trials. So the design, the type of study can be used for any type of intervention that we are testing against some control. It can be in a clinical setting. It can be in a community setting. It can be a behavioral intervention. It can be an educational intervention. We will see some examples where we actually do an environmental intervention. So it's not always clinical interventions. We tend to, those of us that work in the health field, we make the, not mistake, but we make the common assumption that it's always clinical. But clinical is one of the ways that randomized controlled trials are used. They're used in the clinical setting, but they can be used in a more broader circumstances, okay? So randomized controlled trials are considered as the gold standard for establishing that causal association. That's what we're interested in. What causes something between an exposure factor and an outcome. The exposure in our case is something that we control. It's the intervention, and the disease is the outcome variable. And it can be a good aspect of the disease, like getting cured, or it can be a bad aspect of the disease, like getting a death or getting renal failure or something bad, hospitalization for heart failure, for example. So we are interested in that. This is what we would call the basic research question. We also want valid results, okay? To get valid results, now comes statistics to the rescue. Statistics is never about looking at a single case person. Sometimes maybe case series, but rarely also. What statistics does is look at it in a large number of people. It's looking at things in the aggregate. So in order to get valid results from a statistical standpoint, we need large, well-conducted, definitive studies. I don't know if you've heard about the recovery trials, and the recovery trials were started in almost immediately in early 2020 in the UK to study what interventions can be done for COVID at that time. And at that time, there was no known interventions. And so they were looking at the benefit of different therapies and whether that therapy has any benefit or not. And so they looked at all sorts of things, hydroxychloroquine, they looked at dexamethasone, and then finally, once they got the mRNA vaccines, they did huge studies on this. So they did large studies with over 40,000 people in the UK, where they finally were able to show definitively in a well-conducted study across the country that two-dose regimen of these mRNA vaccines gave 95% protection against COVID-19. And this was for severe. It did not prevent you from getting it. It prevented you from getting or reduce your risk of getting severe complications from it. We also want current results. Things change. In every medical field, things are evolving. And as we get more and more information, we need to update what we know of what works, what doesn't work. So and you will see this later with Professor Tambu when we talk about network meta-analysis, that we can do living systematic reviews of what works, what doesn't work. During the COVID treatment, this is no longer active, but the BMJ had online a site you would go to, and you could click on it and look at what works to prevent mortality, or you could select the outcome of interest. It would tell you what is the evidence that exists for different types of interventions at the time. So people want to know what causes things. They want it to be valid. They want it to be current. And so why do we need this evidence? The evidence is needed to change practice, right? Because if we already know that something works, then people will change practice altogether. If people already have changed practice and are doing something, then we don't need to convince them to do it. So to change practice or policy in the country, sometimes we have to take actions. Maybe we have to convince people that something is good or bad for you. Like I can tell you, some people are easier to convince than others on certain things. If I have to convince you that regular exercise and a diet that is low in sugars and moderate in fats is good for you, some of you are going to be convinced. Not every one of you is going to be convinced. Is every one of you going to change practice? Probably not. Have I changed my practice? Probably not. I don't exercise as regularly as I should, but I don't smoke. Some people do smoke, and those people need the governments. We would like to convince them that smoking is dangerous to your health from a heart standpoint, not only a lung cancer standpoint, but we have to convince them. Who do we have unconvincing people? The tobacco companies. The tobacco companies want to make more money. So they don't want the people to be convinced. They want more smokers to be convinced, smokers to continue to smoke, and new smokers to come. Why? I was very easy to convince against smoking. When I was in second standard, we had parents' days where parents would come in and talk to the students and tell them what they did. So a policeman would say what they did, a carpenter would say what they did. And one person came and he was a medical doctor. And he came with a show and tell. He had two lungs in ether. One was pink, one was black. And he said, this person never smoked, this person smoked. I was convinced when I was 7 years old, I didn't have to have a randomized controlled trial to convince me. But convincing people and convincing governments is the hardest thing to do because there are other interests behind this. And we have to think that in mind, that people have interests behind stuff. Because we know a lot of things that are bad for you, but we still have people do it. How many of you drink sugar sweetened beverages? A lot. Pepsi Cola advertises a lot. Thumbs Up advertises a lot. And it's easy. That's one of the worst things for diabetes, etc., and promotes it. So convincing is hard. So we have to convince people. So we need evidence. Now, as I said, if people are convinced, no trial is needed. I was easily convinced, but I was only one case. If the effect is huge, an RCT is not needed either. When Salk came up with the polio vaccine, it worked. I think he had 3 people. I don't remember exactly, maybe, yeah, on himself. His family. He did his family. And that convinced everybody. No trial was ever done on this. You all know that they call it the first trial was James Lind, and this was when the British were going around conquering the world. They would go in ships that would take a long distance to get to places, and the sailors would get scurvy because they'd take lots of meat, they'd take lots of maybe vegetables sometimes, or hardly ever vegetables would go bad, and they would eat fish or whatever, and they would get scurvy. Nobody knew what caused scurvy or how to treat scurvy. And this guy, James Lind, took 12 people. 2 of them got vinegar, 2 of them got seawater, 2 of them got, I don't know, some other stuff, and 2 of them had citrus or limes. And they were the only 2 that got better. And so in a non-randomized control, it was controlled, but non-randomized experiment, he found out this works. And so therefore, everybody was then issued citrus or limes when they went on conquering the world. And that's where, there's an anecdote, that's why the British sailors are called limeys, because of that. But it was this was a randomized, not randomized, this is unrandomized, but controlled experiment. It worked. No other trial was ever done of this. If variability is 0, an RCT is not needed. What do we mean by that? It means that if everybody that gets to an intervention gets exactly the same result, then we don't need it. There's no variability. So if everybody that jumped out of an airplane without a parachute died, I don't need somebody to convince me that jumping out of an airplane without a parachute is dangerous to your health. There's 0 variability. Well, actually, the BMJ had an article about this, that there has never been a trial on whether jumping out of an airplane without a parachute is dangerous to your health or not. So we don't know yet. We should do an experiment. And so they published in 2003, parachute used to prevent death and major trauma related to gravitational challenge. They did a systematic review, and they said our search strategy did not find any randomized controlled trials of the parachute. Well, you laugh because it's stupid, right? Well, actually, you know how these British are with their sense of humor. They published this in the BMJ. Recently, about 3 years ago, somebody did this trial. You imagine it? They went and did this trial. They interviewed people, they recruited people to see if they would be interested in volunteering for a trial to jump out of an airplane without a parachute. Some of them are going to be randomized, to wear a parachute. Some of them were going to be randomized to wear a backpack that had no parachute in it. They talked to something like 70 or 90 people. They got 26 to agree. Can you imagine they got 26 to agree? They did the experiment and they published it in the BMJ, but they didn't tell anybody that jumping was from when the plane was on the ground. They never said you were going to be high up in the air. Okay, and then they showed their results. They had 92 aircraft passengers were screened, 23 agreed. The outcome was composite of death or major traumatic injury. Results, parachute use did not significantly reduce death or major injury. There were 0 in one, 0 in the other, because they jumped from when the plane was on the ground. They must have had informed consent, but nobody asked whether the plane was going to be up or down. They didn't know the height, I guess. I'll have to read the paper more clearly. But this is a joke, right? But in reality, seriously, we have to inform consent. We have to do all sorts of things. But that again? Yeah. Right. And so they said the participants included were on aircraft at significantly lower altitude. So everybody was already there. But those that did not participate were at 9,000 meters, and they also did not die because they didn't jump from there. So I just wanted to show you a joke, but so nobody ever does this kind of stuff. The British sense of humor is kind of strange. But anyway, this was published on the parachute. They call him the parachute investigator study. But now I want to talk to you about serious study. And this is the one that I was talking to you about in the introduction. And this was, again, I call it pure luck. It was a study that was being conducted at the University of North Carolina and 12 different clinical sites across the United States. Our university was the statistical coordinating center, just like Professor J. Asilan said, that CMC is the coordinating center for the polio study and other studies here. We were at that time the coordinating center for the Lipids Research Clinic's Coronary Primary Prevention Trial, CPPT. It was a definitive trial in the sense that in the United States at the time, this is 1971, so in the 1940s and before, people were dying from infectious diseases in the United States. But after World War II, where people died from war, now people were dying from chronic diseases more than they had what they call the epidemiologic transition from infectious to chronic. So people were starting to die more of cancer, living longer, dying more of cancer and heart disease. And it became an issue. You'd go, the doctors still smoked. I remember going to see my pediatrician and he would be smoking as he's looking at it. But when you go to your heart people, my father would go and visit the cardiologist and the cardiologist was saying, no, you should, eat a healthy diet, do this, do that, etc. It was unknown, or it was questioned whether cholesterol was really a risk factor then. Yes, there were observational studies. There were multiple case control studies. There were cohort studies. The Framingham study had been publishing information. But the egg industry, the meat industry, the other oils and fats industry were saying, no, lipids are nothing to do with this. It's because people have these other conditions, genetically this or whatever. And so what this study did is it took people with very, very high levels of total cholesterol, primarily LDL cholesterol. Men, why? Because men are at higher risk for MIs and coronary death, but they had to be free of coronary heart disease symptoms. So they wanted people that had high levels of cholesterol, but already had no signs of that. So it took, for every patient that they recruited, they had to screen 100 people to find one that would meet the eligibility. Their hypothesis was that if you lower their cholesterol, you would lower their coronary death or non-fatal MI. At that time, the intervention was very crude. Cholestyramine, those you guys are all too young to know, this was a horrible medication. It was sawdust, basically, that you would have to take. And just like when you go to the car dealer to have the oil change in your car, what does the dealer have in the floor? He has sawdust so that if any oil comes, it's absorbed, and then he sweeps it away. Well, the same thing here. You take sawdust 6 times a day, mix it with orange juice or water so you could take this junk in. And what does it do? All the fats in your GI system are sucked up and come out with salts. Imagine this. Some people were getting the sawdust with active cholesterol lowering medication. Some were getting placebo sawdust. The pharmacoepidemiologist said, for you to get the benefit of lowering cholesterol, these people need to take it 6 times a day for 7 years minimum. A horrendous thing to do. Imagine if you're getting placebo. Worse than that, cholestyramine would give you either constipation or diarrhea. So they put some medication in the placebo so it would do the same thing so they would not get unblinded. Remember, everybody has to get the same thing. So you have to get the same adverse events. This study probably would not be ethical today. It was done in those days when there was nothing else. So imagine taking horrible sawdust 6 times a day, having diarrhea, horrendous stuff, right? Not getting any better. So this study was, it started in 1971. I was still high school, so I had nothing to do with this study. But I finished my PhD in 1980, and I joined them in 1980. And so I knew at the end what was happening. And I worked with this and worked on the follow-up study of the people that worked on it and so forth. But I got to know what was going on. But this one showed that for every 1% drop in your cholesterol, you got a 2% drop in your risk of a heart attack. This study, when it came out in 1980, was published in 1983 in January, and in March, the Time magazine had it on the cover. Now we, the entire world, worry about cholesterol. And they put a sad face there. These are now the bad news. This is the impact that a clinical trial can have if it's a large, definitive trial. Now, most of us cannot do this huge study. This was $120 million invested by the United States government to do this 10-year study. That in today's dollars would be over $1 billion. So that over 10 crores? Yeah, over 10 crores. So of US dollars. No government is investing this kind of stuff now. What they're doing is, yes, some investment is happening, but it's not at this grand scale. So we don't have the opportunity to do large definitive trials. Sometimes we do. Most of the times we end up doing medium or small clinical trials. But if they're done well, you will hear from Professor Tambu, you can take the information from multiple well-done clinical trials and create evidence from a systematic review or meta-analysis. Yes. So the definitive trial is one that will answer the question and convince somebody, in this case, the government or legislators to make policy. And so it's one that convinces. Yeah, you're right. I mean, what is definitive or not, you're totally correct. It has to do with how hard it is to convince people and what are the forces against convincing, like this Tobacco Institute. for example, for smoking, or the Pepsi Cola for the sugar sweetened beverages, etc. Or the Department of Education that doesn't want to change the curriculum because your evidence showed that curriculum, this one is better than your curriculum. They don't want to be convinced. So those are, yes, but what helps you in convincing is doing it larger. The larger you do it, and the better and the more well conducted it is. If you can show and claim, we did this with good precision. We don't have all this heterogeneity in the outcome. The people, yeah, we showed that we standardized things and we got a strong effect and we got a statistically significant effect as well in a large enough magnitude. If it's a small magnitude, even if it's statistically significant, it's not going to convince people. You want to have a large magnitude, but that requires that you have an idea of this thing is going to work more and by how much. We'll see tomorrow when Professor Jayasilan is talking about sample size, that you really need to estimate what is the effect that I'm expected to see and that I want to detect. What is the magnitude of it? I'm going to get a hazard rate ratio of 1.1. Who cares? I want a hazard rate ratio of 1.5. Wow, is your intervention going to achieve that? That's why when you do a clinical trial or control trial, you don't do that as your first study in that area. You have a lot of information. You know how things behave in observational studies. You have people have done, not you necessarily, but others have done case control studies. People have done cohort studies. People have done cross-sectional studies. There's a lot of evidence that this is likely to work, but there is that ambiguity yet. There's still that inconclusiveness, because if it was not inconclusive, then why are you doing it? So there has to be a little bit of doubt yet, some doubt, but you know a lot about things. The equipoise has to be there. This was the US NIH, National Heart, Lung, and Blood Institute. Yeah, I mean, so if the Indian Council of Medical Research would do it, or WHO would fund it, if WHO had funded this, probably would have impacted worldwide. They were actually doing the Monica study at the same time, but that was a observational study. It was not an intervention. So yes. No, I think not so much. I mean, the Gates Foundation does a lot of things in HIV, and what the problem is with private funders is that as a sponsor, they need to be seen as independent from the researchers, in the sense that we're just giving you the money. We are not influencing you in any way. We're not wanting yes, we would like the result to show that our drug is better, but we're not going to call you at 2 o'clock in the morning and ask you to do this or do that or change that or whatever. They should be independent, right? And so many people question sometimes because we don't know how truthful pharma X was, right? So not all private ones are looked at, but the regulatory agencies, the European Medical EMA, or the Medical Research Council in the UK, or the Indian Council of Medical Research, or US FDA, they look at it more carefully because it's coming from private. But they also look at it, if it's a very controversial issue, the intervention or the outcome, like with HIV, it's, when it's death or not death, or progression from HIV to AIDS. Now there's a lot of patient advocates that are interested. So people will scrutinize your trial if it is of relevance to their condition, or to a condition of their family, or in some places the condition of the country, or the whatever. But we'll be talking about the ideal way of doing clinical trials. We have to realize that in different settings, it may not be, but you still want your trial results to have some impact in convincing relevant people of its importance. And so even smaller, less definitive trials need to be done well. Okay? That's the main message. All right. Why the medical community loves RCTs? Because its relationships are complex. And so we have this thing called confounding. And if you have taken a course in epidemiology, you've heard about confounding. Confounding is when I have a relationship between an exposure and an outcome, but there may be another variable that if I do not include it in the analysis, I get a different result. Right? And if I include it, I get a different result from not including. So there's confounders. So I may show that this intervention is associated with this outcome, but oh, wait a second, maybe if I had included and considered the age of the person, I would have gotten a different result. Maybe if I had included whether they're diabetic or not, I would have gotten a different result. Maybe if I took their gene, did a GWAS, and I found that they had this particular SNP, it would have gone different. So there are so many things. Because relationships are so complex, what are potential confounders? There's many. I work in heart disease, so we usually talk about diabetes. We talk about their diet, their fats consumption, their obesity, waist to hip ratio. All of these things are potential confounders of relationships in cardiovascular disease. But the benefit of this, that we have to control them, and either we control it by design or we control it by analysis. And in the clinical trials, we want to control as much as possible by design. And you will see that one of the processes that we do is that first word, randomized controlled trial. Randomization is the process by which we control the confounders. So that randomization, and here's if you do randomization in a large number of people, then you will get that the distribution of the confounder in the intervention arm will be similar to the distribution of the confounder in the control arm. But that is an if, large sample size. Again, Dr. Jayasilan's talk on sample size will become very important. If it's a relatively small study, you're still subject to confounding, and people tend to ignore it. They said, oh, I did a randomized controlled trial, so therefore I don't have to adjust for confounding. We have what's called residual confounding, meaning confounding that has not occurred. And the residual confounding, we can adjust it for known confounders. What about the unknown confounder? Hey, I don't know, so I can't do anything. If you do a large trial, then not only do the known confounders are balanced out, but the unknown confounders are balanced out. And that's why we like large clinical trials. As I called, small clinical trials, and mentioning the word randomization as, oh, that ensures that you get the balance. I call that insurance is the same insurance that you get from a used car salesman. When they're selling you a used car, they say, oh, no, yeah, we guarantee this. You will get a very good car. That guarantee is the same thing as you get here if you have a small sample size. You want big sample size. You will ask me how big is big. I'll leave that to Professor Jayasilan. The bigger, the better is what all statisticians will say. All right. So the objective then is to understand the relationship in the population. And here comes, from a statistical standpoint, some of the issues. Whatever relationship there is between that intervention and the outcome, it's subject to variability in the outcome of interest. So not everybody that smokes heavily gets lung cancer or heart disease. And not everybody that gets lung cancer or heart disease ever smoked. So there is variability in the outcome. That leads to uncertainty. The relationship is likely impacted by many potential factors associated with it. Those are those confounders. And unless we're doing a huge trial, like the recovery trial with 40,000 people, or the coronary primary prevention trial was only 4,000. Nowadays at the institute where I work at McMaster University, a small study is 4,000. They usually do 15,000, 20,000, because the director of the institute is Salim Youssef, who wrote the paper in 1982. This is why we need large, simple trials. And that's what we adhere to. We try to do large things. But we have this, many potential factors are associated with it. So this residual confounding is likely to give you uncertainty in the result. We recognize also that our observation of the phenomenon in a subset from the population is subject to who we chose in that sampling. The sampling, and we'll talk a little bit more about sampling, is not what we have done, what we do in statistics, where we take a random sample. Why? Because I cannot say to my patients, you were randomized to be in my study, or you were selected to be in my study, you're going to get this intervention or not. You have to consent to participate in my study. So now I have to inform you of what it's all about, and you have to make a decision. You self-select yourself. In statistics, that sometimes, for a nice way to say it, we call it a convenience sample. Or the correct way to say it, it's a garbage sample. It doesn't work for statistical analysis. The only way that garbage sample works is because we randomize. If we don't randomize, then it is not statistically a good enough to do any statistical. We're just too polite and we call it a convenience sample. But that gives you, if I'm studying people that are in the United States who participates in clinical trials, are usually the poor people, because they don't have health insurance. There's no universal health coverage. So if they participate in a trial, then they get seen by the doctor. And so their trial may be for who knows what, for diabetes, but now it's saying, doctor, I have this pain over here. They'll treat that pain too. They'll get good health care. So, but maybe their other conditions are different. So now, is it really generalizable, this result, to the general population? Yes and no, right? Because there are idiosyncrasies of people that are in very poor, precarious conditions that participate in these things. In other places, you have all sorts of people there. So you want sampling may give some sort of uncertainty, but doesn't invalidate the trial, because you still have the distribution of people similar at baseline between the two arms. Fourth thing is recognize that any model that attempts to explain the phenomenon is subject to misspecification. We're going to do a simple model, which is exposure and outcome. And maybe we'll do some models where we adjust for some residual confounding. But maybe it's not a linear model. Maybe it should be a nonlinear model. Maybe then, is it the correct relationship? Maybe the response of the intervention is not at a higher dose, I get a higher response. Maybe there is a threshold that it goes at, there's no response here, then it goes a response there, and then it plateaus out. So then there is, whatever model I'm using, I have to maybe misspecify. And finally, recognize that what may be observed may be due to chance. And this is what statistical significance comes in, and testing it. The key word, can you guess what the key word is? No? The thing in red, uncertainty. Everything is due to uncertainty. So we will have to worry about this uncertainty. And that's what we try to minimize the uncertainty. We can't get rid of it totally by designing a study that is large, that is well conducted, where we're standardized, where we control things so that we don't have uncertainty playing a major role in it. Now, we're going to be quantifying relationships, and depends how we ask the question, what we're looking at. So is blood pressure associated with BMI? Okay, so yes, so we have two continuous variables there, we can do a correlation measure, etc. But if we have, is blood pressure associated with obesity? That's a very different question, because obesity is yes, no, obese versus not obese. And so I have a continuous variable blood pressure. There is no correlation measure here. And we can do some graphs on it. But is hypertension associated with obesity? Now, hypertension is binary, and obesity is binary. So now I have two binary ones. So now we do have another measure of association called the odds ratio. If it's in a cross-sectional study or a case control study. But is obesity associated with the time to becoming hypertensive? Now, and I guess here we could have done relative risk if it was a cohort study. But see that people say obesity, or I'm looking at blood pressure and BMI, but I'm actually interested in the time to becoming hypertensive in people that are not hypertensive. So now it's a time to event variable, and we have the hazard rate ratio over time, and we have different measures of association. The point of this slide is just to say that when you formulate your research question, you have to be careful of what is the variable that you're studying. What is your intervention? Here it's maybe reducing blood pressure or lowering people from hypertensive to normotensive. What is it that you want? You want to lower the blood pressure by 10 units? Or you want to 10 milligrams, or do you want to lower it by 20 or what? So you had a question? No. So when you formulate your research question, and that's one of the exercises you'll do today, talk in the afternoon about it, you have to be careful what are the variables that you study. Because according to the variables, you will have different measures of association, like we have over here, and the design will I mean, the measurement of it will be different. Yes. You are better take care, not always 100% take care. Okay. Yes and no. You design your study, and we'll talk about it when we determine the sample size, that the first thing you do is you determine what is clinically important. The statistician will not do that. You as the clinician will say, this is a clinically meaningful difference. Now, based on that, we would say, what is the necessary sample size to be able to detect that or a bigger one? And we will give you the necessary one, not 4 times the necessary. So we will not give you an overpowered study, etc., because you don't want it. Because if you get a very large sample size, it's exactly what you're saying. You will get a clinically unimportant thing being called statistically significant. I also, just as a semantics issue, I don't like to use the word significant outside of statistical context. So the word significant, please reserve it for statistical talking. When you talk about anything else, you say clinically important, clinically fantastic difference, clinically awesome difference, clinically meaningful difference, whatever, but not clinically significant, because then people drop the clinically and then we don't know what's happening. So clinically important, so something that is clinically important is not necessarily statistically significant. And something that is statistically significant is not necessarily clinically important, for the same reason he mentioned. If I have a very large sample size, then anything unimportant, non-meaningful, can be shown to not be due to chance. And that's what statistical significance is showing, whether it's likely to be explained by chance or not. And you may have a small study that has a fantastic result that shows you a relative risk reduction of 40%, or it shows you a hazard rate ratio of 1.8, but you did it in 500 people, and now it's not statistically significant. So you may have a very clinically meaningful result, but you cannot say that it's not due to chance. So we have, this is the care in the design of the sample size. It's very important. But it starts not with the statistician telling you the sample size. It starts with you telling the statistician, this is what I think is clinically important. And then we go on from there. Oh, okay. The larger, yeah. No, that's right. You're correct. Exactly. Be physically significant. Yeah, you're right. Yeah. So I was just talking about a confounding standpoint, but not necessarily for the significance part. Very. Oh, that is there a measure of association between a continuous variable and a binary variable? No, there's none. Between two continuous variables, we have a correlation measure. Between two binary, we have the odds ratio. I missed the relative risk here. And if we have a binary and a time to event, we can have the hazard rate ratio over time, or if it's constant over time, we have a single hazard rate ratio. But if you have one is continuous and another one is categorical, there isn't a measure of correlation or of association. So we can look at it, we can model it, but there's not a number that we get. Yeah. Tough question, but I would lean towards what you said. Ethics will drive this. Why would I subject a large number of people to show that this intervention is going to lower your blood pressure by 1 millimeter of mercury if you do this? It's balancing is the issue here, right? You don't want clinically unimportant stuff to be detected necessarily. You want to have adequate power to detect clinically meaningful things. You don't want to be necessarily overpowered because that's unethical from the patient's standpoint, but also the whoever's funding you standpoint, because now you're taking money away from another study. Yeah, I mean, if there's a lot of variability here, we can control that by how we do things. But if there's you say biological variability, a lot of noise, what we tend to do is our eligibility criteria for the trial tries to restrict the types of people so that we don't have all of that biological variability. That's why that trial only took men 35 to 49, 59. It did not take any women. Mm-hmm. Well, I mean, so hypertension medication is usually not tested with superiority trials anymore. We'll talk about that. So we'll talk about the different types of hypotheses. So because there is medication that works for that condition, when you get a new intervention, whether it's a new pharma company developed intervention or whatever, they're not going to try to show that this is superior to what's existing. They also are not trying to show that they cannot show that this is superior to placebo because it's unethical to give placebo to people. So they tend to do, and we'll talk about this, the non-inferiority hypothesis and where that comes from. So they want to show we are not so much worse than the existing standard. And then there, the sample sizes are big because the differences are small. Yeah, so this is for association. We're looking at correlation. If I wanted to look at effect, difference in differences or difference in means or differences in medians, we could do that. So that's the next slide. And then we could do regressions. So we want to claim that a relationship is causal. And so causal means if and only if. If the exposure is present, so if the intervention is present, then the outcome occurs. And if the outcome occurs, then the exposure must be present. So that's the if and only if part of it. So we establish causation, and there's multiple ways of doing it. We, in this course, are going to only talk about the causation by design, and that's when you do randomized experiments. And that's, as I said, you basically are doing a true experiment as much as possible where you control everything and change one thing. You may come across other types of terminology that's used. We won't use it here because it's for observational studies. These are terminology primarily used in psychology, sociology, in a lot of other disciplines that do not do as many randomized controlled trials. And so they will do these sorts of modeling with mediation analysis or structural equation models or direct acyclic graphs. These are things beyond what we're going to talk about in this course, but we'll talk about the true experiment. And so they will say, what are mediations and causal pathways, and they'll do all sorts of things, but we are not going to talk about it. Our causal evidence is going to be based on doing the experiment. Now, we always have complications in experiments. So we need to make sure that the groups are the same at baseline. So when we are randomizing to one arm or the other, there is an objective way of randomizing or allocating people. So we talked about no allocation bias. It should be objective. Never have the investigators do it. Never have the patient select what they want. And you want to randomize large numbers of individuals so that if there's any confounders, they are distributed the same. We have that inherent variability of the outcome was another source of uncertainty. And that if there's a lot of variability, for example, blood pressure varies all the time and so much. But death from all causes, any cause, that doesn't vary. You're dead or you're alive. That's why a lot of studies look at the outcome, death from any cause. There is certainty. If you want death from coronary heart disease, wait a second. Now there may be some uncertainty because somebody may have died out of hospital. There isn't a death certificate. They died suddenly. Oh, well, that was probably heart disease. Maybe, maybe not. Who knows? Maybe it was a brain aneurysm. And bam, they're gone. But we tend to attribute it, if they're over 60, we tend to say, oh, yeah, it's with heart disease. There's uncertainty there, right? And so we standardize the eligibility criteria to make sure that there is less, we restrict, as you were saying, we restrict who comes into the study so that everybody's homogenized. It's not heterogeneous. We don't take wild range of ages. We then take wild range of condition. We exclude people that have, if we're studying heart disease, we exclude people with renal problems, we exclude people with cancer, we exclude people with HIV. Why? To reduce that heterogeneity. Because yes, those are things that's reality has heterogeneity, But for our study, we are making it very artificial. The randomized controlled trial is pretty artificial because we do all of these things. Okay? We standardize the eligibility criteria. We standardize the procedures and the measurement of the outcome. So, we tend to do these things where we have the outcome is adjudicated. There is, and we'll hear more about this tomorrow, that there is a committee that looks at all the medical records of all the participants that had the event. If my event is heart failure hospitalization or it's coronary death, and they'll they follow a standard criteria to determine, looking at the hospital records and this, and if the person died, they do verbal autopsies from the family or the doctors, and they get all sorts of stuff. And then they say, oh yes, this person, no. Following the same criteria and doing it objectively. That is a big issue. To standardize, that is to reduce the noise. Heterogeneity is basically noise in the system. Why? Because sometimes the signal, which is the effect of intervention, is small. If we're comparing a new intervention against placebo, it's likely to be a big effect. But ethics dictates that we compare in a superiority trial the new one against standard of care. And unless I've been lucky enough to develop a new intervention that's going to get me the Nobel Prize, I'm going to have a small effect. And that small effect is a small signal. So if I have a lot of noise, those of you that know about engineering, we talk about the signal to noise ratio. If the signal is large, it doesn't matter what noise there is, I'll hear it. But if the signal is small and the noise is a lot, forget it. I'm not going to detect it. That's what we're trying to do here. We want to reduce that noise. So despite having a small signal, as long as it's clinically important signal, I can detect it. If it's a small, unimportant one, then who cares? Why am I doing the study in the first place? And then we had the confounding, and we already talked about confounding as a complication. Now we also have post-randomization confounding. Once we randomize somebody into the study, now they always forget, they're no longer, we think of them as participants, but they continue, believe it or not, to be humans, right? And so humans do whatever they want to. When you go to the physician because you have some infection and the physician tells you, okay, yeah, you have to take this antibiotic for the thrice a day for the next 7 days. What do? You sometimes you forget, sometimes you take it. Oh no, wait a second, no. My sister had some antibiotic. I'll just take that. I won't buy this one. And I'll take that one. Not the same one. It's a different one. Oh, she only had 15 tablets. Oh, I'll just take 15 tablets. Or you start taking it. After 2 days, you feel good, and you stop taking it. Or after 2 days, you feel bad, and you stop taking it because you feel bad. Who knows what you do? You don't follow things. People don't follow things. You have to realize that in your study, those people are also humans. They don't follow. They've signed consent. They said, yes, doctor, I will follow this. They will do exactly like any other human. They will not. So you will have poor and different types of adherence. Oh, I'm getting nausea. I think they gave me this pill. Or I'm not getting nausea. I think they gave me placebo. So I will do this. I will change my behavior because of the way I imagine what they gave me. Or I'm lost to follow-up. Why? Because when I have to go to the clinic every 2 months, there's no stupid place to park. And they don't give me money for transport. So I'm not going back. Or because I'm feeling better, I'm not going back. Or because I'm feeling worse, I'm not going back. Or because the administrative assistant insulted me last time, did not offer me any coffee, I'm not going back. Who knows what people do, right? So they are lost to follow-up. And if it's related to the outcome, then it's worse. You have missing data, data of poor quality, you have to train people. So all of these things we'll talk more about tomorrow. But these are parts of things, when you're designing a study, you need to think about humans participating in the study. And so people will behave differently. They're not guinea pigs or animals that you put in a cage and do a study like that. They will not necessarily follow them. And these things may lead to bias. If people that are feeling poorly because this medication causes nausea stop taking it, then they're not getting the effect of it. You will see that we are forced to analyze the data as if they were taking it. We call that intention to treat principle. And we'll mention more about the intention to treat principle tomorrow. But that will weaken the study signal. And we want the study signal to be strong because we've designed it. People follow the protocol, then we should get this signal. So RCTs are at the top of the research evidence hierarchy. We all know that. At the bottom is expert opinion. This is this usual practice. You know, in the old days, Dr. X would say something and, oh, yes, if you say it, I will follow. That nobody does that anymore. If we look at case series, a bunch of people that have this and what happened to them, yes, you can describe it. You can get details. You can do time series, you'll see it over time. But then we do observational studies. Observational studies can be cohort, case control, or cross-sectional. They give you different types of evidences and association measures of how much this is related to that. So you get prevalence from one, you get incidence from the other, you get strength of association, and you can do meta-analysis of observational studies. Originally, meta-analysis, as Professor Tambu will say, were designed only for randomized controlled trials. Nowadays, we're doing it for all sorts of types of studies. There's more noise in those, so they're difficult to interpret, but people still do them. But we then have above the line, we have now things that are experimental studies. We have the quasi ones where there's a hint of causal association. We'll talk about those. Then we have the randomized, so we do get causal. And then finally, meta-analysis and systematic reviews of randomized controlled trials. That's sort of at the top of the pyramid, so to speak, of the research evidence. Now, all statistical studies are on a sample from the population. So we have the universe. So we have here, this cloud is the universe, unobservable world. There are parameters, they're of interest. We have the population here. We take a sample, we calculate something similar to whatever we're interested in the measure of association, for example, or the effect. I could have put an effect here. And then we get the probability sampling enables statistical inference. So now from the observable world, we do statistical inference. In a clinical trial or experimental setting, what we have is we do have now the same population here, somewhere there. We have the effect size, we have the relative risk reduction, we have the hazard rate ratio in the population, the unobservable world. We take self-selected volunteers. There's no sampling. That's the garbage part. And then we have a garbage sample, but then we have a process of randomization into intervention and control. That enables us to get the effects or the signals, which are the relative risk reduction estimate, hazard rate ratio estimate, or the effect size estimate. And then statistical inference enabled us to tell us about this in the population. And this is what we do in randomized controlled trials. I did not put the word garbage here, but I should have put the word garbage in there. But that's what that is. But the process of randomization is very important. So research questions are very important. And as I said before, the way you think about your research question will determine how we design your trial. So for example, if I say, what is the association between physical activity and obesity in children? That leads to an observational study. I'm just looking at an association in a group of people. So I'll have a target population. I'll take a sample from it or from the sample population and then get that. While if I ask the question is, what is the effect of a physical activity intervention on obesity in children? Now I'm experimenting. So I'll still have the same target population, but now I have, instead of a sample population, I have a study eligible population. And I will take then the volunteers, and the process of randomization is what is needed to be able to do statistical inputs. Okay. So the research question is important. And I want to sort of finish off here with the research question. It's not a simple matter. Okay. There's a great paper here by a colleague at McMaster on posing the research question. It must be well framed, reflecting clarity of the thought process, and it determines your design, right? Your research architecture, strategy, and method. The way we talk about it for clinical trials or controlled trials is the PICOT framework. What is the PICOT framework? The P stands for the population. That's determined by the eligibility criteria. This is your target population. The intervention is your exposure. The control is the comparator exposure or comparator intervention. The outcome is the variable on which you will judge the effect of that intervention. And time frame, multiple time frames, is duration of intervention as well as duration of follow-up. Many people say, oh, this is a 2-year study of cataracts. And I'll show you one. The cataract intervention took 3 minutes. Follow-up was 2 years. But the study the intervention is 3 years. Or that CPPT one, what was that? That was a study that took 3 years to recruit, and minimum follow-up was 7 years. So is the study a 7-year study, or is it a 10-year study? Whatever you want to call it, but say something. Don't just say this was, a study with that. It's a 7-year study or 10-year study. And then the setting. Was this done in rural communities? Was this done in tertiary care hospitals? Was it done in highly specialized clinics that had the latest in technology and the best trained physicians? Or was it done in community hospitals that had this or that? For example, the setting is very important. So in this paper on the research question, it says the step 1 is identify the research problem or idea, consider options for the PICOT element for framing the question, focus the research question, and then check whether the question follows the finer criteria. And what is the finer criteria? Finer is feasible. Is it feasible to do? So you have to do a pilot study if you don't have pilot information from others, or get collaborators, see at the cost of it. Is it interesting? If nobody cares about it, why are you doing this? To get your PhD degree or to get a thesis done? Is it ethical to just do it for that reason? Is it novel? Yeah, somebody else hasn't done it. That would be important. Because if it's done already, then why are you doing it? If the question is answered. Finally, is it ethical? To include the ethics approval prior to conducting is very important. And then, is it relevant to what? This goes back to convincing people. So is it going to be just to scientific knowledge, or is it going to change policy? Is it going to change how people treat patients? If it's not going to do anything like that, then why are you doing this? Is that research question important? So today in the afternoon, you will be developing a research question. And so you will be having to think about these characteristics of research questions. So there are good things, bad things, and ugly things about clinical trials. So it can establish causal relationships if done in a true control experimental fashion with standardized protocols in a homogeneous set of units. So you have very high internal validity. You will know if this intervention works or not. And it deals with known and unknown confounders if you randomize a large number. But it's artificial. Like all experiments in our chemistry lab, they're artificial. All experiments that we do in our physics lab are artificial. This is another artificial. It's not real life. Because in real life, you don't limit the population. You don't standardize things. People do whatever. And the interventions are not administered in a standardized way. So we usually do these in a highly specialized clinical center, usually in high or middle income countries with resources. So the results don't necessarily apply in less sophisticated settings or with general population. They have what we call poor external validity. And the ugly part is that in order to have the good part, they cost money. And they're complex to do. They take time and effort. Many of you are going to be doing clinical trials as part of your career. How many clinical trials will you be able to do in your productive life? If you're doing a trial, it'll take 3 to 4 years of your life. You're going to work 30 years? Are you going to do 10 clinical trials? You have to do studies that are prior to clinical trials. You may do 4 or 5 clinical trials. And you want to design them properly. You want to have a proper research question that is important to do. Not many people are going to do tons of trials. They'll do a few trials in their life. And so you're dedicating your life, your career to doing some of this. Yes, answer important research questions. That's worthwhile investing part of your career to do. We'll talk more about this later. There's different types of trials. I'll skip this part. We talk about because that's on the next lecture, this will come back, so I'll just skip this. We'll talk more about it. There's efficacy trials on infectious diseases and chronic diseases. We have what we call pragmatic versus explanatory trials. And the more pragmatic a trial is, it's the more applicable to a variety of settings. That's the way to think about it. A pragmatic trial is applicable in a broad place setting. That's why I always like to put the S for setting, because that is very important. If you do it in a highly specialized clinic that only less than half of a half percent of people in the country are going to benefit from this, is it worthwhile the investment of your time as well as of whoever's sponsoring you? You want things to be more applicable to a broader sense of people. Those tend to be more pragmatic studies, are more important from a public health standpoint to a larger number of people. And those are actually harder to do than to do the highly trained ones. So the explanatory ones are actually easier to do, but they're not as important in their impact. The ones that are more impactful are the harder ones to do. We will talk about different types of control studies. Changing people's behavior is the worst thing to do. It's the hardest thing to do because human behavior is hard to change. But surgical trials are relatively easy. Pharma, drug trials are relatively easy. It's the ones that are people have to do something for a period of time. Those are the hard ones. And then to get them to do it after the study, it's even harder. So there's a way of describing them as explanatory or not, how you do it, but we'll talk a little bit more about that this afternoon. But beyond RCTs, this is sort of the introduction of RCTs. And I wanted to just mention that, yes, we'll talk only about RCTs here for the next lectures, but don't forget there's other things. You will get systematic reviews and meta-analysis at the end of the week, but there's also meta-regression. Is when we have residual confounding that we cannot control by design, that comes from small trials or clustered trials, pragmatic trials, and observational studies. Despite doing meta-analysis, we still have a lot of heterogeneity. There is things that we're not going to cover in this course, but that you know about, meta-regression. And then many people think of RCTs as a religion, okay? And people get very rigid about it, and they discount other kinds of evidence. It's an important kind of evidence, but it's not the only kind of evidence. Okay? So you can get other evidence from quasi-experimental studies as well as from observational studies. Or, as I said, when I was in second standard, from just looking at it, impressing a kid. and get results. Okay. And as a joke, I want to finish off with beware of the religion of trialism. So trialism is any other religion always has a holy trinity. And so here we have the clinician, the patient, and the biostatistician. This was actually published in 1978 in biomedicine. But it's so that, and so by the patient, God is perceived as the physician. The clinician perceives p-value less than 0.05 as God, and the biostatistician, according to this, perceives themselves as God. But there's also a devil in every religion. And so the devil by the patient is to be randomized. They don't want to be randomized. The clinician is a p-value greater than 0.05 is the devil. And the biostatistician is a sample size that's very small, n of 1. And so they also have 10 commandments. And the 10 commandments are, thou shalt randomize is number 1. Number 2, thou shalt double blind. Number 3, thou shalt not do unto a patient that which thou would not do unto thyself. Ethics. Number 4, thou shalt commit consecutive patients. You will not do that sampling thing. Number 5, thou shalt follow the protocol. Number 6, thou shalt complete all clinical forms. Number 7, thou shalt use appropriate statistical evaluation. Number 8, thou shalt seek answers to only a few questions. And we'll talk about multiplicity of secondary outcomes and subgroup analysis. Number 9, thou shalt not adulterate the data, no tampering with the data. And number 10, thou shalt not take the FDA or the Medical Research Council or whoever's name in vain. They're the ones that, if you're in pharma, you want to follow it. But anyway, sorry about that joke. All right, more questions?